Good morning. Welcome to a new EBD seminar series. And today our speaker is Chris Liedtke. He's Juan de la Cierva postdoc at the EBD, at the Wetland Ecology department. And Chris, he did his PhD at the University of Basel about sexual differentiation of African amphibians. And since 3 years ago, he came to the EBD, and now he with different postdoc contacts. So he's gonna do the last seminar of this season that has been a little bit different with this online, with this pandemic situation that has forced us to do online seminars. And also Chris is gonna help us for the next year for the coordination and organization of the new season of the EBD seminar series. So thank you so much, Chris, for coming here. It's great that you are doing the last one. Like, it's a super beautiful way of closing the seminar series. And also I would like to do some take-home messages. Some like small messages. The first one is if you need a certificate of attendance of this seminar, please write your name in the chat of the YouTube channel where the streaming is going on, and also send us an email to seminarios@ebd-seq.es, and we will send to you as soon as possible. We are trying to finish all the certificates this week, so apologize for the delays of the other seminar certificates because we have been really busy. And also write your questions in the chat of the YouTube channel, and then at the end of the seminar, August Camacho, he will ask the questions to Chris. So thank you so much, Chris, for giving us a seminar, and you can start talking.
Okay. All right, great. Thanks, Irene, for the introduction. Yeah, so last seminar of the session, so no pressure, right? So yeah, I'll spend hopefully around 40-45 minutes talking about amphibian life histories, evolution at different scales. And this is something I've been working on over probably the most of my research career this far. Okay, so variation is this concept that obviously is important in a number of different disciplines in biology, but it's particularly important in evolutionary biology. In the most simplest terms, we could even define evolution as evolutionary processes, so selection, drift, acting on variation. And through this rather simple and elegant concept, we get sort of this unfathomable biodiversity that we see literally everywhere around us. So one aspect of this diversity is the differences in life history strategies. And by that, I refer to sort of age and stage specific patterns and timings of events that make up an organism's life, and they can be anything from growth rate, age of sexual maturity, but also things like the number of offspring or the type of mating systems. And these traits obviously vary a lot between species. So the lifespan of different cephalopods, for example, can vary tremendously, but despite the Humboldt squid's rather short life, females produce many more eggs than a Pacific octopus can in up to 5 times that lifespan. But sort of these life history traits, so they don't only vary across species, they also vary within species. So tarsiers, for example, within the same species can live as either monogamous or polygamous groups, depending on resource availability. And fire salamanders throughout most of their range give birth to aquatic larvae, but then there's like a handful of populations in northern Spain that give birth to terrestrial juveniles. So we can take that one level deeper even, so we can look at variation within individuals of the same species. So this, for example, refers to things like the length of hibernation of dormice that can be different from one year to the next, depending on the severity of the winter. And it can be things like alpine newts that can either live completely aquatic lives, reaching sexual maturity, but sort of retaining larval features like the external gills, or they can undergo metamorphosis and instead spend most of their lives or at least a great portion of the year, out on land as these terrestrial adults. So if we come back to that definition of evolution, to really understand why we see so much diversity in life history, we have to understand sort of this diversity at different scales. So we have to understand the phylogenetic patterns across many different species that are shaped by these evolutionary processes, but we also have to understand the mechanisms that are capable of creating this variation within a single species or even closely related species. And today I want to talk about 3 interconnected projects that kind of fall within that framework. And so my talk will also be divided into 3 parts. So a major driver of evolution is obviously selective pressures that are imposed by environmental factors. So first I want to talk about an example where specific environmental factors have been important in shaping the evolution of life history, specifically reproductive strategies. And then how these strategies or individual components, or sort of the individual components of these strategies are inevitably intertwined and interconnected. And so we also have to understand these connections and how they might influence this diversity. And of particular interest here has been the actual size of the organism's genome. And that's what I'm going to talk about in that second part. And for both of these, we're only going to focus on sort of explaining phylogenetic patterns and what has shaped these differences across species. So then for that final part, I'm going to shift gears a little bit, and I want to focus on the molecular mechanisms, specifically gene expression that can create this variation. And I'm going to look at developmental rate as the life history trait, and how that can change in a single species, and how this variation can scale up across closely related species. So as the title already gave away, all of these studies are going to focus on amphibians. And so hopefully by the end of this talk, I'll also have convinced you that amphibians make this great system for studying life history evolution. So in no small part, why amphibians are so great is because they're the tetrapods with the most described diversity in reproductive modes. And most of this diversity is actually found in frogs. So these are Anura. And most of my talk will actually be looking at examples within Anura, but not exclusively. So I thought right off the bat, I'm going to also point out that there's 2 other groups of amphibians, the newts and salamanders, which I suppose most of you are also familiar with. But then there's a third group that's often forgotten. These are the caecilians. These are legless amphibians, many of which live underground, and they're restricted to the tropics. So that's probably why they're less known, at least in the northern hemisphere. So in terms of life cycle, the Anuran plesiomorphic life cycle is often referred to as being biphasic. And what that essentially means is that you have aquatic larvae that hatch from eggs that are placed in water. And these larvae undergo metamorphosis to become morphologically, physiologically, but also ecologically really different, distinct adults. And in many cases, these are also terrestrial. So because of this life cycle, amphibians tend to be highly dependent on aquatic habitat. But this dependence varies. So this plesiomorphic life history reproductive mode has actually been heavily modified across evolutionary time. So for example, not all species lay eggs in water. There's many examples of species that either carry around their eggs or they lay their eggs in specialized foam nests. And most common, I would say, are frogs that stick their eggs onto vegetation. But these eggs sort of still give rise to tadpoles that are still dependent on some pool of water where they continue their development. But then there's also cases where these tadpoles, these larvae, are actually highly adapted, almost terrestrial larvae. So there's cases like the poison dart frogs where they're sort of semi-terrestrial and the adult can carry the tadpole from one pool to the other. And then you have sort of substantially more terrestrial tadpoles, like ones that have sort of develop hind legs very quickly and can hop around wet rocks. And you even have cases of fossorial tadpoles that live underground. Then there's the species that have completely lost the larval stage. So these, I think I would group into two categories. So you have what's called direct developing species. And here they lay eggs usually on land, but sometimes in these specialized pouches like these marsupial frogs. And they don't have a larva. So from the egg, you get a tiny little mini frog that sort of hops out with no need for metamorphosis. And sort of a derived version or a similar version of this would be viviparity, where the developing embryo is actually carried by the female. So this has evolved independently in the 3 different orders of amphibians. And what's quite interesting is that this is actually really rare in frogs, but rather common in caecilians. So that's also another aspect of how this varies across the amphibian tree of life. Okay, so hopefully from that, it was clear that sort of a major axis of the diversity of these reproductive modes depends on aquatic habitats for reproduction. And it's this sort of terrestrialization or this evolution of independence from these water bodies that I want to focus on for this first part of my talk. And this is a project that I was working on primarily with Simon Loader and Hendrik Müller. And the focus was on a specific group of African amphibians. So when we look at global distribution of terrestrial reproductive modes, we actually see that they're not uniformly distributed. So terrestrial reproducing amphibians tend to be more concentrated in the tropics. And I think that's quite nicely shown on this map here by a study by Lyon et al. But already, for the better part of a decade, there have been studies that have shown this and have specifically shown that there seems to be a strong correlate with temperature and precipitation. But even when you look inside, or sort of just within the tropical biome, you still see that these frogs or these amphibians are not randomly distributed. So there's still sort of some structuring. And in the 60s, this idea was proposed that, well, I mean, okay, these terrestrial frogs, they're more terrestrial on paper, but they're actually still highly dependent on humid environments. So these terrestrial eggs actually need to be in very moist, usually sort of foresty undergrowth. And so this hypothesis emerged that, well, you still, like, you're going to find these things in wet, moist forest habitat, and that's what's driving the evolution of this reproductive mode. More or less at the same time, though, there was sort of this non-mutually exclusive hypothesis that said, well, okay, yes, moisture is important, but what's really driving the evolution of this reproductive, this shift in reproductive modes is the topography. So in these steep montane forests, you don't actually find, standing bodies of water. So eggs and tadpoles have to be adapted to extremely fast-flowing streams, or better yet, reproductive modes become completely independent from the necessity of these water bodies. So those were the hypotheses that we were going on. Is it climate, is it habitat, or is it topography that's shaping the evolution of terrestrial modes? To test these hypotheses, we used phylogenetic comparative methods. So this could broadly be defined as using evolutionary relationships of species to pose or test hypotheses on evolutionary processes. And the building blocks of such an analysis would essentially be understanding the evolutionary relationship of species, which we can do by constructing phylogenies. Nowadays, they tend to be molecular phylogenies. And then on that phylogeny, we can map traits. So these can be discrete characteristics like reproductive modes, but they can also be continuous traits like body size, number of eggs, things like that. And of course, they can also be extrinsic factors like precipitation or temperature. So we decided to focus on African members of a single family, the bufonids, the true toads, and that was based on a couple of reasons. So they're kind of a neat system because they're found basically across the whole continent, spanning different habitats and climate zones. And within that single clade or within that single group, we've actually got most of the reproductive modes that I mentioned earlier covered. So we have the typical aquatic breeders, we have sort of specialized aquatic breeders that lay their eggs in small pockets of water, like tree holes or even water-filled snail shells. So these big African snails. And then we have these stream breeders that have tadpoles that are adapted to living in fast-flowing water. So these tend to be tadpoles that have big sucker-shaped mouths that allow them to stick onto the rocks. And then we have more terrestrial modes. So these would be species that lay their eggs on land, most of them in this case also being direct developing, and then truly viviparous frogs as well. So actually viviparity has only evolved a handful of times in neurons, and both only sort of the extant remnants of this evolutionary innovation is restricted to this clade. So they're not found this reproductive mode in frogs is not found in any extant species anywhere else in the world. So we reconstructed the most complete phylogeny for this group. We had about 80 species, which is around 78% of the described species at the time. And then we mapped the reproductive modes that I just detailed onto these and reconstructed ancestral states. So what we found was that the different terrestrial modes tend to evolve independently, usually from aquatic ancestors. And what I thought I'd point out here is that you can see quite nicely that these closely related species tend to have the same reproductive mode. And this is why it's really important to include these phylogenies in these models, because when you're comparing across species, species seldomly tend to represent statistically independent data points. So that's important to keep in mind to not violate these model assumptions. Okay, so we had the species and reproductive modes, and now we wanted to know, well, can we characterize the habitat or the environment that they live in? And to do that, we used remote sensing. So we compiled a data set of around 8,000 locality records. And then using those locality records, we extracted 6 environmental parameters. So we had 2 parameters describing climate, temperature, and precipitation, 2 for habitat, so the percentage forest cover, and humidity, and then also 2 measures of describing topography. So the first was slope, and the second is something called the topographic wetness index. And that's essentially a measure of whether or not you're expecting water to drain into an area, in which case you're more likely to find aquatic habitats in that grid cell. So obviously those are all really intercorrelated. So we calculated species means for each of these 6 variables, and then subjected them to rigid rotation by a PCA. And so here I'm just showing the first 2 components of that PCA with the species colored according to their reproductive modes. So hopefully you can see that the x-axis actually does a pretty good job at distinguishing these more generalized blue species from the more terrestrial modes, as well as these yellow stream adapted modes that are showing up on higher values of that first PC. I think that's even more clear when I overlay the groupings that we got from a hierarchical clustering analysis. And what you can see here is that the terrestrial breeders tend to cluster in the same cluster, and that's characterized by steeper slopes, more forest, and higher humidity. We actually tested that statistically, so we constructed binary models where we had either aquatic or terrestrial reproduction as the response variable. And then, and with those models, we found that the first principal component, as well as the topographic variables, are actually highly significant. Next were the habitat parameters that were marginally significant, and we actually found no effect of climate. Okay, so when we talk about these reproductive modes, what we're actually talking about is sort of this orchestra of traits. So they're made up of many different things. So direct developers, for example, generally tend to be much smaller. They tend to lay only a few eggs, but these eggs are quite large and they take a long time to develop. It contrasts that to these biphasic breeders that can lay, tens of thousands of eggs, and these eggs usually take only a couple of days, maximum a couple of weeks to develop. So these individual traits, body size, egg size, developmental period, they all tend to be quite tightly linked. And so to understand the diversity in life history, we can't only focus on extrinsic traits, but we also have to look at how these interactions between these traits are impacting each other and what effect that has on the diversification of these, on the diversity of life histories. So that leads me to this next part, where I want to talk about a study where we looked at different components of these reproductive modes in more detail. So specifically, we're interested in understanding how genome size might be related to these life history traits. And that's something that we did here at EBD. So I worked with Juan Gomez-Mestre on this, but we also had collaborators at the Natural History Museum in London, Dave Gower and Mark Wilkinson. So to provide some context, genome sizes are yet another trait that varies hugely across the tree of life. And amphibians are actually the group of vertebrates with the largest difference in genome size. So yet another good sort of model system to work on for questions like this. Again, there's sort of so many hypotheses out there on what exactly is driving genome size evolution. And one that we thought was particularly interesting was this idea that, so we know that large genomes impose a constraint on cell replication rate. So large genomes take much longer to replicate. And so it was hypothesized that this would actually scale up and it would actually impose some sort of limits on the maximum developmental rate of an organism. So this is particularly relevant to amphibians because these biphasic species, they undergo this immense remodeling and development in that short space of time when they go from tadpole to frog. So these biphasic species were hypothesized to actually have smaller genomes when you compare that to direct developing species that don't have that sort of tight constraint during metamorphosis. So it was thought that there would be less sort of selection against large genomes. So in order to test that, we collected genome size information for amphibians of all three groups of the frogs, salamanders, and caecilians. So most of that information was made available through this online database, but we also measured our own genomes. So we selected species with to specifically targeted to sort of improve our sampling of the different reproductive modes. We measured, I think it was around 60 species ourselves. And then we mapped these genome sizes onto a published phylogeny. So here I'm showing that phylogeny published by Pyron, and the ranges and the medians of the genome sizes. And what you might be able to see is that the genomes for salamanders are actually substantially larger than for the other two groups of amphibians. So we can reconstruct the ancestral genomes of these internal nodes on that phylogeny and then basically show that same information in a slightly different way. So here I still have time on the x-axis, but the phylogeny has been distorted so that you have genome size on the y-axis. So I think now it's way clearer, genome sizes of salamanders are absolutely gigantic. And then what we did for each of the three orders separately is we coded species as either having larvae or not. And then we performed a phylogenetically corrected ANOVA to test whether whether there's differences between these reproductive modes. And we actually found that there wasn't. So for none of these three groups, we find a significant difference. And actually, although the trend that genome sizes are expected to be larger in directly developing species was there for salamanders, but we actually saw the opposite for frogs. Okay, so reproductive modes themselves don't seem to pose a constraint. But what about the components of these reproductive modes? I mean, even within the biphasic mode, we're seeing this huge variation in developmental period. So could there be something there? So we wanted to dig a little deeper. So in addition to this genome size information, we also broke down the reproductive modes into 3 of its parts. We looked at adult body size, the diameter of eggs, and the developmental period, which we defined as the time it would take to go from egg to froglet. Obviously, we can't completely ignore the extrinsic factors, especially for amphibians. So these are obviously poikilotherms, so they're much of their physiology and metabolism is directly impacted by things like temperature, for example. So we can't get rid of that completely. So when we have these complex systems of a number of different interconnected traits, a nice way to try and model that is using path analysis. So the idea of path analysis, I think, is fairly intuitive. So you construct a hypothesis about how you think these variables are interconnected, and then you display them by using these directed acyclic graphs. So in this example, for example, I'm saying that climate and egg size affects developmental period, and that this developmental period then in turn affects genome size. So climate and body size also have an effect on genome size, but this effect is actually indirect. Then under the hood, basically you're running a set of regression analyses. This may not be the only hypothesis you have. It wasn't the only hypothesis we had. And for example, we thought it was also reasonable to assume that if egg sizes are correlated to cell sizes, then we'd also expect there to be a direct link to the genome size. On the other hand, you might have sort of a more null hypothesis where you're saying like, okay, all the life history traits are connected, but actually they have nothing to do with genome size. So I think it's also important to test that hypothesis. And another sort of great or tool or sort of a nice thing about these path analyses is that they can actually infer causality, or at least try to. And you can do that by just flipping that arrow in that first scenario. And now, instead of saying that developmental period affects genome size, you're actually saying genome size affects developmental rate. So in total, we came up with about 20 scenarios that we thought were reasonable. And we ran each of these models, always correcting for phylogenetic non-independence. And then just like you would do for any other kind of model selection, you'd calculate we calculated an information criteria, in this case, something called the C statistics, but the behavior is basically the same as the Akaike information criterion. That's probably more well-known. And then based on that, you can essentially rank the performance of these models. And then you're either left with a best model or a set of best models. And then those best models can be averaged. And that's what we did. So for frogs specifically, this is what that average model looked like. So here is the hypothesis that we started off with, and then the thickness of the arrow showing the size of the standardized regression coefficient, blue if it was negative, and dashed if it was non-significant. So if if it overlapped with 0. And basically what that's telling us is that there is a strong link between genome size and developmental period, but that developmental period is also affected by these extrinsic factors, by climate and this aridity index. So from that, it seems like at the macroevolutionary level, so at the level across species, we're seeing that these 3 aspects, developmental period, genome size, and climate, are all tightly linked. Okay, so I think I've kind of raced through those first 2 parts. But essentially what I wanted to do here is recap at what it was that we're actually trying to do, which was looking at patterns in the processes that are shaping life history diversity in amphibians. We could show that sort of the environment obviously imposes certain restrictions and can be the driver that generates this diversity. So specifically, we found that topography, not just climate, might be very important in shaping probably one of the most striking evolutionary events in amphibian life history, which is the evolution of terrestrialization and the loss of the larval period. But we've also seen that intrinsic factors, specifically the size of an organism's genome, can have a great impact on different components of these reproductive modes, specifically the developmental period. So up until now, I've only really addressed these broad patterns, but we haven't really talked about these molecular mechanisms that are actually able to create this variation that evolutionary processes can then act on. And that's what I want to spend the next part of my talk on. So if we go back to this slide from just a second ago, I had shown you that developmental period is actually correlated with environmental parameters across species. But everyone working with amphibians obviously knows that developmental period also varies within species. And so understanding this variation within species is also really important. And that's something that we did. So this was a project that I did with Juan here at EBD, and in collaboration with Ewan Harvey, who was in Liverpool at the time. So to try and answer this question, we looked at a single species in particular, which was the Western spadefoot toad, Pilovetes cutripes. And this can be found throughout most of the Iberian Peninsula, and it's a biphasic species, so it breeds in aquatic habitat and has a tadpole phase. This species really likes seasonal ponds, so it's quite common in Doñana. But as you may know, these ponds dry up in summer. But the different ponds, they dry up at different rates, and the rate at which they dry up also changes from year to year. So you have this sort of mosaic of extremely heterogeneous environment. And in such an environment, it's quite advantageous if you're plastic. So if the tadpoles of the spadefoot toad can actually adjust its rate of development so that when they sense the ponds drying out, they can actually accelerate their development and reach metamorphosis early. And that way they can avoid certain death through desiccation. So Juan and colleagues have actually quantified this plasticity. So they took the tadpoles and brought them into the lab and standardized conditions such as temperature, day length, and things like that. And they could show that by simulating this desiccation, so by dropping the water in the aquaria that these tadpoles were in, you could actually accelerate their development by 30% or more. What's really neat is that we have a closely related species in North America, or in central North America, Scaphiopus cauchi, which has this hugely different developmental rate. So again, this is a species that breeds in these ephemeral ponds, but these are more like puddles. These are pools that, some of them dry up in a couple of days. And accordingly, the larval period of this species is also extremely short. I think one of the shortest in amphibians. So they go from egg to frog in, the space of a week or two. Compare that to Pilovetes that takes 3 to 6 months. I also wanted to point out here that these two actually sort of fit perfectly into what I had just talked about, in that the genome of Scaphiopus, the fast developing species, is less than half the size of the genome of Pilovetes that takes much longer to develop. So it's not only that absolute larval period, but also their plasticity that's hugely different. So when you perform that same experiment where you simulate desiccation, Scaphiopus doesn't actually change its developmental rate anywhere near as much as Pilovetes. So again, this is all stuff that was done before I joined the group here, and we know a lot about the system. So we know, for example, that there's 2 very important hormones that are driving that are sort of cueing these developmental processes and even metamorphosis. Thyroid hormone and corticosterone. And what they showed was that the endocrinological levels of both of these increase as a response to desiccation, but they're constitutively high in Scaphiopus. Not only that, so when you're confronting these species with the stressor, you see Pelobades increasing its metabolic rate. It burns through all of its fat reserves. It delays gonad maturation and reduces its size at metamorphosis. And all of these traits tend to be much less plastic in Scaphiopus. So interestingly, Scaphiopus generally, sort of the Scaphiopus phenotype seems to represent the accelerated morph of Pelobades cultripes. So in benign condition, these two species have very different life histories, but then they converge when Pelobades accelerates its development. And why I think this is particularly interesting is something that I want to come back to right at the end. Okay, so this is where I came in. So we wanted to see what's actually driving these changes at a molecular level. And for that, we set up, we wanted to look at differential gene expression because we wanted to characterize the genes and that network that's underlying this developmental acceleration. And we also wanted to see what happens to the expression of these genes and that network when you're comparing between these two species. So we can do that by setting up an RNA-seq experiment. So again, we took tadpoles from both of these species, we had them in standardized conditions, and then we dropped the water level on some of them. And that way we could extract RNA from control and treatment, and then sequence it using short read technology. So for those of you that are unfamiliar with this concept, basically the RNA that's been extracted is then enriched so that you're left with mostly mRNA. So these are genes that are being actively transcribed. And then that RNA is fragmented, usually into pieces of about 100 base pairs, translated to DNA so that it can be sequenced. And then those fragments can be mapped back onto genes. And the number of fragments are proportional to the starting amount of RNA. And that's basically allows you to have a relative measure of the expression of some genes compared to others. And more importantly, for our purposes, we could look at the expression of the same gene in different conditions. So obviously, with this transcriptomic approach, you don't just get information for a single gene, we get information for thousands of genes. And we also didn't just want to look at the effect of treatment, but we also wanted to see how this effect differs between the species. So to kind of try and wrangle all of this information into something more tactile, we applied this technique called weighted gene correlation network analysis. And that's essentially a clustering technique that groups these genes into modules based on the similarity of their co-expression across samples. So to try and visualize that, this is the dendrogram that you would get from this clustering approach for all of these thousands of genes. And then we can cut that dendrogram into modules. So in our case, we're left with 8 modules of genes that behave more or less in a similar way. And I mean, these modules still contain thousands of genes, but what's really neat is that we can now condense this information into eigengenes, which are essentially first principle components of the module expression. So from this mountain of data, we've now got sort of a much more manageable, structured, biologically informative set of modules with their eigenexpression values. And we can just use these eigenexpression values per module as our response variable and then plug them into GLMs, and then have species and treatment as our predictor variables, and also the interaction term between these two variables. So here I'm going to show just some examples of those results. So for example, that large turquoise module that you saw in that dendrogram, that was composed of nearly 8,000 genes, and it had a significant species effect. So what that means is that these genes are expressed at different levels in the different species, but their expression is not dependent on the water level. So these could be termed as genes whose expression have evolutionarily diverged along with the divergence of these species. And when we look at the function, the biological function of these genes, what networks and pathways they're involved in, we see that they're mostly related to things like mitotic cell cycle and tissue remodeling. So they might have something to do with the much faster development in general that we're seeing in Scaphiophus. Interestingly, though, we also found a module that had a significant species treatment interaction. So that's really cool, because basically what that's saying is that these are genes whose expression is responsive to the stimulus, but not in the same way across the two species. So we're seeing here that they're basically non-responsive for Scaphiophus and highly responsive for Peloides. So these genes represent genes whose plasticity has become canalized or fixed over evolutionary time. And when we look at their biological function, we see that they're related to pathways linked to lipid metabolism, metabolism of steroids, and cholesterol biosynthesis. So that kind of feeds back nicely to what we know about the physiology of that system. So what we were able to achieve with this study is we could show that the molecular workings underlying the changes in the life history between these two species, and how that differs under different environmental conditions. And we could do that at the transcriptomic level. And I think more importantly, we could show that this variation in phenotype can scale up across species, and all that's needed to happen is that the sensitivity of these gene networks changes. So that sensitivity becomes fixed or lost in our case. So why that's particularly exciting, I think, is because it means that phenotypic or developmental plasticity might be a mechanism that can create evolutionary diversity. And this is something that's been called genetic accommodation. So the principle here is that you have this plastic ancestor, similar to Peloides in our system, that can change its phenotype or its development based on the environment it's in. And then the descending lineages from this ancestral species, if they start to move into more homogeneous environments and this plasticity is lost, that can actually result in a diverged canalized phenotype. Okay, we made it. So if you're only going to remember a few things from this talk, I'd want it to be this. So variation in life history exists at different scales. And to fully understand how this variation originates and how it's maintained, I think we have to work towards a framework that tries to incorporate these different scales. And that ultimately means integrating different fields or specializations in biology. So for example, comparative phylogenetics can tell us a lot about the patterns in evolution and the underlying drivers, especially the environmental drivers of this, and also the interaction of intrinsic components. But these patterns tell us little about the actual mechanisms that generate this variation. And this is where transcriptomics is really powerful, especially when you're working with non-model systems. So finally, I also want to sort of leave you with this idea that a source of variation need not exclusively be random mutations or recombinations, but they can be processes like genetic accommodation, where I think it's quite an exciting, potentially controversial topic, because it means that environmentally induced phenotypic changes can precede changes in the genotype, which I think is quite an exciting avenue in evolutionary biology. So with that, I want to say thank you. Thanks to all the members or all the people that have been directly or indirectly involved in these projects over the many years. Thanks to you guys for listening, and a big thanks to Irene, Mari, and Agus, who've been doing a fantastic job with this seminar series. Thanks.
Okay, thank you very much, Chris. It's been a really rare pleasure to observe your presentation. It's absolutely exquisitely presented. I love all the graphs and the whole process of deduction that you followed from patterns to processes. I really liked it. So we have to start with, we have a couple of questions that have been raised by Carles Villar. We'll proceed to read them now. So the first question from Carles is the following. thank you very much for a very interesting presentation. You say that faster development leads to smaller genomes. This makes sense to save energy while trying to invest in fast development. So, okay, this is the first part of the question only. So it makes sense to save energy while trying to invest in fast development. However, is it advantageous to have large genomes in a species with a slow development? Is it advantageous to have a large genome if you're going to develop slowly?
Yeah, so yeah, I guess I only sort of touched on sort of very relevant aspects of these hypotheses. So the idea behind this hypothesis that's been postulated since, so I think it was in the 1950s when people started comparing genomes across species, and what they realized very quickly is that genome size is not correlated with the complexity of organisms, and actually most of the weight of the genome comes from the non-coding regions. So the idea was that these non-coding regions can just keep growing in size. So there's some studies that are showing that insertion mutations are more frequent than deletion mutations. So they could essentially just keep growing. And if there's nothing that's impeding that growth, or if there's no negative effects of that growth, you would have genomes getting larger. And the minute you have some selection or some driver that would favor a reduction in genome sizes, that's when you would start seeing a reduction in these genome sizes. So that's the hypothesis, and I think that's where this idea of related to developmental periods comes from.
Very interesting. So we have more questions from Maria Jose Ruiz Lopez. Thanks for a great talk. And you said that aridity and mean annual temperature influence the size of the genome through the effect of developmental rate. Could you clarify what is the direction of that relationship?
Yeah, so if maybe I can go back to that slide.
That would be great.
There? Yeah, so so the direction, I'm not sure if I follow entirely. So I think I
can repeat the question. Do you want me to repeat the question?
Yes, please. Sorry.
Oh yeah, sure. We have a problem with accent here. So Mari says, you said that aridity and mean annual temperature influence the size of the genome through the effect of developmental rate. Could you clarify what is the direction of that relationship?
So, well, I mean, I could talk about the aridity index. So the aridity index, for example, is a measure of, if I remember correctly, so the higher the index, the more humid the environment. So the reason why we were interested in including that was specifically to try and test whether the sort of the length of the hydro period of ponds could impose restrictions on how long species have in the larval period. So I have to say I have to go back to the paper now. I'm not sure if I'm exactly answering what Mari is asking. So the direction, as in, I have to go back to the paper. Sorry, Mari.
No worries. Mari has a...
No, I was writing in the chat. So it is, yes, yeah. For example, I'm looking at mean annual temperature. It has a direct blue line with genome size. So what I'm wondering is whether higher mean annual temperatures tend to have bigger genome sizes, for example.
Yeah, let me, after the seminar, I can try and send you the...
Okay.
Sorry, yeah.
No, it's okay.
No worries. I was following on this line. I was also curious to know, why did you choose mean annual temperatures and not other descriptor of annual temperature?
Yeah, that's a good question. I think there's always a debate about that. So for example, a lot of people working with amphibians, they like to choose the temperature in the wettest quarter or wettest month. And the reasoning is that that's the time of the year that amphibians are most active. And I think that works well in tropical zones, but it doesn't work so well in temperate zones, because sometimes you get areas where the wettest period of the year in terms of precipitation is actually in winter. So when you have a lot of snowfall. So then that doesn't accurately reflect the temperature that frogs or amphibians in general are active. So we tried a few different measures, and we settled on mean annual temperature as being sort of the most universally applicable or meaningful variable. But I agree that there's a lot of debate about that.
Yeah, I was thinking that maybe the genome size and the developmental period might have like some sort of different being subjected to different aspects of temperature. Like, for example, genome size being affected by a cumulative effect of how many generations have passed across the history of the lineage, that you could have more in hotter environments. But at the same time, having hotter environments might tend to affect aridity and then somehow reduce developmental periods. So maybe including a couple of temperature variables might help you. Some sort of different thermal variables that can represent different types of selective pressures, for example, over genome and over developmental period.
Yeah, totally. And I think you make this point really well that I've been trying to make. That, I mean, I think it's really important to really look at these things at different scales, because I mean, very often, I mean, there's especially more recently, there's a lot of phylogenetic comparative methods that have been developed that include some measure of variance, but usually you're left with a single value that you're representing a whole species with. And we know that these things change over time, they change across geographic space. So I think that's why it's really important to look at all of these layers. And so, for example, today I didn't talk about the levels, sort of the between across species and within individuals, but that's also something that we've been looking at. So we've been looking at two different populations of the spadefoot toad, one from Madrid and one from Doñana, that are experiencing very different conditions, and they have done so for a long time. And we want to see if we can, these patterns that we're seeing in gene expression going from Pelobades to Scaphiopus, whether we're also seeing them when we look across different populations.
Very cool, Chris. I have a couple of more questions are piling up in the chat. So now we have one from David Pablo Quevedo. He says, as I understood, it seems like the genome acts like a great USB, and the more information it receives, the more bigger it is. So it could be possible that in fast development, the individuals could suffer any maladaptation due to a lower quantity of information about the environment.
Yeah, I mean, I think it's a really exciting thing to look at, this genome size, because I mean, so we've been looking at genome size as if it was sort of a physical trait, so the physical size of the genome has an impact on other aspects at the cellular level, but also the organismal level. But there's also a lot of studies that have tried to look at things like the actual information stored in the genome. So there was quite a nice study by Katharina Wollenberg, I think 2016, where they tried to see if there's sort of larval specific genes, so whether the genome of biphasic species have the burden of having to carry all of these extra genes or regulatory regions required, to program all of these developmental steps that direct developers might might have lost, for example. So I think it's really exciting. I think a major drawback, or sort of something that is slowing us down still, is that amphibian genomes are really, really large. So even the frog genomes that are modest compared to the salamander genome, many of them are larger than the human genome. And they also have a lot of repetitive elements, which makes it very difficult to sequence and assemble the genomes. But I think we're getting there. I think soon we'll see a big push in that direction.
Okay, but do you see any that having a large that in populations with fast development, you think that the genome size could be a maladaptation? Could be because the environment is giving less information? I think that adjusts a little bit more to the question of David. So David wants to know if the individuals could suffer in fast development. Could the individuals suffer any maladaptation due to a lower quantity of information about the environment? I'm thinking that he wants to know if the genome size could be negative for them in a situation of fast development.
No, I mean, so there's models that show that the larger the genome, the more likely you're going to suffer a deleterious mutation. So in that sense, there's at least the hypothesis that larger genomes would be more at risk in that sense. But in terms of missing information, I mean, I don't know if there's any evidence for that. I mean, I could be wrong, but I think, I mean, my reasoning is that the number of genes, for example, is really constant across organisms. Mm-hmm. So, I think vertebrates tend to have around 30,000 genes, and that doesn't change. Obviously, we know that there's a lot in the non-coding sequences that affects how these genes are being expressed. Whether information is stored in those non-coding regions that is then lost, and that's sort of maladaptive, I think is an interesting question, but I can't give you an answer on that.
Thank you. But I think that answers perfectly David Pablo's question. Carles Vila has another question in the line on the conservativeness of genome size. So he says that among vertebrates, genome size is much more variable in amphibia and fishes than in mammals or birds. Could genome size be less under control in ectotherms? Are there like less regulatory systems for genome size or something like that? Is no?
Could be. Yeah, could be. I'm not sure. I know that so within amphibians, the gigantism in salamanders is kind of a hotly studied subject, and it seems like there was a mutation that led to sort of a malfunctioning retrotransposable element controlling enzyme, so that the repetitive elements just went absolutely wild, and that's what led to the large genomes. I think most of the hypotheses are based on this principle that genomes will grow unless there's something that reduces the size. At least that's my understanding. I'm not sure if I don't know enough about the mammal and bird genomes, but it would definitely be interesting.
Okay, thank you very much. So we have a lot of questions here coming, a lot of interest here. Irene Mendoza comes with another question. She says, according to your results, is there a pressure for having less plasticity of traits as evolutionary processes take place?
Yeah, that's something that we always talk about in our group. So we tend to look for examples where you have like a plastic state becoming fixed, but that has to happen the other way around too. So I think that's really interesting, and I can't think of many cases where that is. Actually, that's something that I think would be really neat. So yeah, I think interesting, but can't tell you more about it.
That is something. Yeah, that process of phenotypes coming from plastic to conservative or to constitutive is coming along with several evolutionary studies like this one. Yeah, that's a really interesting point. Okay, so Guillermo Belo Anton has another question on part 1. He says, do those African toads use small water bodies or puddles for reproduction? If so, I guess this could not be included in the analysis as you only refer to standing water bodies.
Yeah, so we did code them. So we do have, if I can load that slide. Here, so we do have species like Nectophrinae that breed in tree holes, and Mertensophrinae that, I mean, people have found tadpoles in cattle hoof prints, so just sort of small depressions filled with water. So we did code them, but I have to agree that, I mean, I think it's very hard to, probably impossible to really capture that at the level of remote sensing, because obviously, I mean, especially in places like tropical Africa, you know, the best resolution you get is something like 1 kilometer by 1 kilometer grids, and these, you know, these water-filled tree holes, I mean, they're this size, you know. So yeah, I mean, they're there. We tried to code them, but that's definitely a caveat that it's very hard to capture that, I think.
Okay.
I mean, I'm sure. I mean, it must be similar with the salamanders in northern Spain. So I wonder if, because there's this hypothesis that I guess Guillermo has worked on extensively, that you know, the viviparity in salamanders might also have been driven by lack of standing water. And I wonder if you guys have come up with a good way to characterize habitat in that way.
He says, Guillermo says, we found a steep topography related to the distribution of viviparous salamander lineages, salamandra. However, we cannot account for the water bodies, as these species can also use puddles to the liver's larvae.
Yeah. Yeah, I think, I mean, I think it'd be really useful to be able to find some way of getting a remote sensing estimate. For example, with the tree holes, we thought that, you know, percentage forest cover would be a good estimate. But yeah, in cases where these species breed in depressions, or like I said, I mean, these Mertensophrinae toads, I mean, they've been found inside water-filled snail shells. And yeah, beats me. I've thought about you know, using some soil type maps and, you know, permeability of the ground to estimate puddle formation, but that still wouldn't capture, you know, other water bodies, like anything else that can catch a bit of rainwater.
You know, I am doing that actually. I have, well, they are running right now at the supercomputing of Cesca, modeling exactly the hydro period of different types of ponds across Northern Africa and Europe. And well, we still have to see. In a previous occasion, we had the opportunity to how you say, to evaluate the accuracy of these models, and they work well for a couple of ponds. But I don't know. I still don't know for across a big geographic range. But yeah, it's actually possible to do that using niche mapper. So in case you want to talk about that, guys, you want to try and see if this new characterization of water availability is interesting, we can talk about it later.
Yeah, definitely. I mean, I think the key would be that it's able to go down to like a ridiculous size, you know, like exactly less than a meter, ideally.
Centimeters.
That's amazing. And then the other thing is like, you know, if that information was available on a temporal scale, so you would, you know, if it's only filled for 1 week, I mean, that might be highly relevant for some of these amphibians, for example.
Yeah, the idea is to try and identify in which places, taking into account the type of soil and other stuff like slope, etc. How long can you form puddles?
Yeah, I mean that would be really cool. I mean, I think, you know, inevitably these will always be sort of the best approaches given the resources, but you know, I think that's the aim of science, right? To try and get as close as possible to what's really out there.
Yeah. Okay, so Guillermo says, we thought about using remote sensing tools. It's still hard to account for those tiny puddles. Exactly. Yes, exactly. Okay, we need some brainstorming. Truly. So I'm absolutely open to see if something of what I'm doing is useful for you guys. All right. I don't see. Does anybody have more questions from the people from the audience? We have almost 20 people there. Okay, if not, I have a question about why did you choose these certainly fascinating examples for studying the gene expression, the spadefoot toads, but why only 2 species? Like,
yeah,
like you know exactly what happens.
Yeah, I mean,
it's
so
gonna be difficult now.
I could, you know, I mean, if you go to like you know, an evo-devo conference, for example, they would tell you like, wow, you did it with that many species. So
I know.
Definitely, you know, for ecologists, you know, 2 species is ridiculous. But yeah, I mean, you know, obviously cost is a huge factor. So these methods, I mean, they're coming down in price, but they're still really expensive. Then, for example, I didn't go into any of the details about the nightmare of mapping genes across the 2 species. So actually, you know, in the end, we had a list of orthologs, and yeah. But I mean, so I think the idea we have. I mean, so this was kind of like something that we wanted to try with the most extremes in this group. But you know, essentially, we want to move towards filling in that gap. So we want to look at try and get a gradient rather than just 2 extremes.
I mean, I think that you can always study what happens across different organizational levels within each species, which is actually what you did. It's just the comparison that becomes difficult to infer anything about it, right?
Yeah.
Between the 2 species. But yeah, I understand. I mean, I did almost the same when I studied my leaders in the PhD. At some point, it's very hard to find like a lot of independent lineages that do this or.
Yeah.
Oh, I hope that you can follow up with that. And one thing that you raised that I thought it was very interesting is this philosophy of trying to start integrating studies from different fields, different specialties. How are you coming along with reviewers about that? Like, are they coming nicely, or are they making questions like I'm doing, like from the stand of ecology or from the stand of?
No, I mean, I think we've had very understanding reviewers, but they might have been reviewers more from the side of you know, differential gene expression studies where they're more understanding, or at least, you know, they understand the practical implications for doing a project like that. So far, no, but it's, you know, it's a moving target. And I mean, so for example, for that study, we had 3 biological replicates per species per treatment. And again, you know, like for most of us, it's ridiculous to talk about means calculated from 3 data points. But you know, I mean, this is what we have. And I think, you know, in a couple of years, you know, especially as costs start coming down, people will try and obviously have more statistical power by increasing sample sizes. So I think it's coming along, but yeah.
You're opening the way, certainly. Thank you very much, Chris. So, well, I have to, if I don't see any, do you? So Carles is thanking again to you and to the organizers of the seminar series, and that has been great. And I hope September we can start with seminars, with presential seminars. Thank you to you, Carlos. Yeah, I hope so too. And I want to thank a lot to the other organizers, Irene Mendoza and Mari Ruiz. And I'm very sorry to say that it might be we're gonna start missing Mari among the organization of the seminars. I mean, it's so hard for me to say this. I hope that it's actually not true in the long run. We will come to you to question about how to organize this.
Yeah, I'll help you.
Something new about it.
Last seminar. Chris is starting to help us, so I'm looking forward to the new season of EBD seminars. I'm sure you guys are going to do a great job, and we will have fantastic seminars. So,
thank you very much, Mari. Irene, do you want
to say?
Thank you,
Mari.
Any last words to farewell this last session of the seminars?
Yeah, so it has been a big effort for all of us. I think that maybe it's not so evident how much work all these seminar series have for preparing the organization, and also looking for the speakers, and then all the communication part. And so I think it has been a lot of effort, but I am very proud of the result. I think we have done very well for the seminar series this year, despite the problems we have. Also, we have, with the support of Sandra and Carlos Ruiz, also
exactly.
It has been easier. And thank you so much also, Carlos and Sandra, for your help. And hopefully we will have another exciting and compromised team next year. I'm not sure if I'm gonna be, but for sure someone else will come and take over us. So thank you so much, guys, for being there and for all your support. And see you in October, hopefully.
Thank you very much to everybody. Bye-bye.